In this video, we're going to get you started using Swiss PDB Viewer. This is a program you can download from the internet and use on a Mac or a PC to view, analyze, and compare three-dimensional structures of proteins, DNA, or RNA. In this video, we're going to show you where to find and download structures to look at, how to open them, how to center them on your screen, and then how to manipulate them, to move them around on the screen like this, to zoom them in and out to look at details or the big picture, and to rotate them. Where do you find structures? You can find structures in the Protein Data Bank. Just Google PDB. Now, the Protein Data Bank also contains DNA and RNA structures and complex assemblies containing both RNA and DNA. This one right here is a crystal structure of a protein bound to a DNA. To get the structure, go to Download, select PDB text and just click, and this will download the file to your computer. Open your structure as usual by going to the File menu and select Open PDB File. PDB refers to the format of the file. If you have a different format, use one of the other commands. When you first open your file, you may not see the structure. This is because it is not properly centered. So just go to the icon in the upper left-hand corner of the program and click on it, and you will see your structure appearing centered in the middle of the page. Three main ways of manipulating the structure are to move it back and forth, to zoom it, or to rotate it. You get back to the original position using the centering feature. The mouse controls all of these manipulations, but what the mouse does is controlled by which of these three icons is selected. You can select these manually, like this, or by using the Tab key to toggle between them. We will complete this video by telling you about the windows that allow you to manipulate the structure. You've already seen the display window, which is where the structure is displayed. By clicking on it and using your mouse, you can manipulate the structure as we just saw. The toolbar window, accessible if you can't see it from the window menu, has those icons we told you about and several more that control other functions for manipulating and for displaying different information on the screen. The layers window tells you which files are open and which are visible. Here we have 5 different files open. The one that is 1YNW is the active one, and it is the one that is visible. You can control which structures are visible in this box, right, this column here. Finally, the control panel over on the right controls which parts of the structure are visible. If you select one residue, you will only display that residue. If you select a chain, you will display the whole chain. And you can change the coloring by clicking on colors and selecting a particular color to use. You can display everything by holding the Shift key and selecting the entire structure. By selecting each chain in turn, we can color each chain a different color. And this helps in seeing how the structure is put together. In this structure, we have 2 chains that make a DNA duplex, colored yellow and green, and we have two proteins binding to the DNA. One is red and one is blue.